Hello everyone, myself Vikrantandan Shive. Today we are discussing about citric acid cycle or TCA cycle. Tricarboxylic acid cycle or TCA cycle. Okay, tricarboxylic acid cycle is also called as Krebs cycle. The TCA cycle is depends on oxygen. It is an aerobic respiration process. TCA cycle takes place in mitochondrial matrix. In this cycle, the first product is citrate, that is 6 carbon compound. So this cycle is also called as citric acid cycle. The TCA cycle is a 6 carbon compound citrate is sequentially broken down and release the energy in a form of ATP and NADH. Steps involves in TCA cycle or tricarboxylic acid cycle. Step number 1. The product released from glycolysis pathway, that is pyruvate, is converted into acetyl-CoA by the enzyme pyruvate dehydrogenase. The 2 carbon compounds of acetyl-CoA combines with a 4 carbon compound oxaloacetate to form citrate. The reaction is catalyzed by the enzymes citrate synthase. This is the first regulatory stage in a citric acid cycle. This step is irreversible and does not require energy. Step number 2. In these steps, the citrate is converted into isocitrate by the enzyme action of aconitase. During these steps, the intermediate cis-aconitase is formed. This is the reversible process. So see here the figure. Okay. So the citrate enzymes, okay, that combines these two molecules of acetyl-CoA, okay, to form compound called as this citrate, that is the 6 carbon compound. Okay, 6 carbon compound. Okay, the next step is this citrate is converted into isocitrate by the action of enzymes aconitase. Okay, aconitase enzymes that convert citrate into isocitrate, but there is an intermediate is formed that is called as cis compound is called as cis aconitase. Okay, cis aconitase. Okay, so then this isocitrate get converted into alpha ketoglutarate as the action of enzymes isocitrate dehydrogenase. Okay, during this process the NAD plus is utilized and the release of NADH. Okay, this is the third number steps. This alpha ketoglutarate is converted into succinyl-CoA by the action of enzymes called as alpha ketoglutarate dehydrogenase complex. Okay, in this reaction the NAD is converted into NADH. Okay, during this process the CoA group is released and the release of carbon dioxide. Okay, the next stage is succinyl-CoA is converted into succinate. Okay, during this process also the CoA group is released. This is the one of the important steps where catalyzed by the succinyl-CoA synthase. It utilizes the GDP and inorganic phosphate to synthesize the GTP molecule. Okay, in these steps the one GTP molecule is synthesized. Okay, the succinate is converted into fumarate by the enzymes action of enzymes succinate dehydrogenase. Okay, succinate dehydrogenase. During this process, the FAD is converted into FADH2. Okay, the fumarate is converted into malate by the action of enzymes fumarase. Okay, and after the malate is converted into oxaloacetate by the action of enzymes malate dehydrogenase. During this process, the NADH molecule is released. See here, there are how many NAD molecule is to be released, that is one here. Okay, then after here second, here third. Okay, then one release of one FADH2 and the release of one GTP. Okay, so moving to our presentation. Step number 3. By the action of enzymes isocitrate dehydrogenase, the isocitrate is converted into alpha ketoglutarate. This is the irreversible steps where NAD plus is utilized and synthesis of NADH. Step number 4. The alpha ketoglutarate is converted into succinate by the enzyme succinyl-CoA synthase. The utilization of GTP takes place. This is the irreversible process where the CoA SH group is released. Step number 5. The enzyme succinyl dehydrogenase convert succinate into fumarate. This is the irreversible process where FAD is converted into FADH2. Step number 6. The fumarate is converted into malate by the action of enzymes fumarase. During this process, the water is released. This is the irreversible steps. Step number 7. In these steps, the malate is converted into oxaloacetate. This reaction is catalyzed by the enzymes malate dehydrogenase. During this process, the release of NADH+. In the TCA cycle, it produces 2 ATP, 8 NAD plus and 2 FADH2. Note, the oxaloacetic acid is not available for next citric acid cycle. That's why there is a multiple pump present on the mitochondria to take the oxaloacetate within the matrix. Okay, thank you.